The immune cascade is a series of events and reactions that occur within the immune system in a response to an infection, injury, or other immunological change. The immune system is a complex network of cells, molecules, and processes that work together to defend the body against foreign invaders such as bacteria, viruses, and other pathogens. When the immune system detects the presence of these invaders, it initiates a coordinated series of actions to eliminate the threat. It can broadly be classified into two main branches: innate immunity and adaptive immunity. Innate immunity is the first line of defense and includes immediate, non-specific responses to infections or injuries. It is present at birth and encompasses four types. The first is physical and includes the skin, which prevents pathogens from entering the body. In addition, it also includes the mucous membranes that line the respiratory, digestive, and urogenital tracts. These structures secrete mucus to trap and expel pathogens. The second is physiological barriers. The most obvious is the gastric acid found in the stomach, which helps to kill off ingested pathogens, although anyone who's eaten week-over leftovers knows it's by no means perfect. It also includes enzymes found in tears, saliva, and mucus, which can break down and inhibit the growth of microorganisms. Finally, it includes antimicrobial peptides. These are small proteins with antimicrobial properties found in bodily fluids. The third is cellular barriers, and these involve white blood cells which eliminate microbes that enter the body. Phagocytes, such as neutrophils and macrophages, engulf and digest pathogens. Natural killer, or NK cells, target and kill aberrant cells, such as those virally infected and tumorigenic cells. Killing is mediated by cytotoxic molecules, which are stored within secretory lysosomes, a specialized exocytic organelle found in NK cells. The fourth is cytokine barriers. Cells which have been infected by a virus start to secrete interferons, which prevents the spread of infection further. Thus, in this way, they protect the non-infected cells from infection. In addition, the innate immune system involves the complement cascade. This is a complex and highly regulated series of enzymatic reactions that play a crucial role in the innate immune system's defense, as well as in other immune and inflammatory processes. It is made from a group of plasma proteins produced in the liver, which help to destroy pathogens. The complement system is composed of a group of proteins that interact in a cascade-like manner. It is named complement because it complements the activity of antibodies and other components of the immune system. The complement cascade can be triggered by various stimuli, including the presence of pathogens such as bacteria and viruses, as well as immune complexes and damaged cells. Complement proteins such as C3b can bind to the surface of pathogens, marking them for phagocytosis by cells like macrophages and neutrophils. The process enhances the efficiency of pathogen clearance. It results in the release of inflammatory mediators such as anaphylotoxins C3a and C5a, which promote vasodilation, increased vascular permeability, and aid in the recruitment of immune cells to the site of infection or tissue damage. Complement proteins can assemble into a membrane attack complex. These are C5b through to 9, that inserts into the pathogen's membrane, forming pores and ultimately leading to cell lysis and destruction of the pathogen. The complement system can help clear immune complexes, which are formed when antibodies bind to antigens on pathogens or other particles. The complement cascade is triggered by three distinct pathways. The first is the classical pathway. This pathway is initiated when antibodies, specifically IgM or IgG, bind to their target antigens. This binding triggers a series of complement protein activations, ultimately leading to the formation of the membrane attack complex. Next, we have the lectin pathway. This pathway is activated when pattern recognition molecules called mannose binding lectins, or MBLs, bind to specific carbohydrates on pathogen surfaces. And finally, we have the alternative pathway. This pathway can be activated directly by pathogen surfaces without the need for antibodies, as well as via injured tissues. It is considered the more ancient and immediate response mechanism. All three pathways converge at the activation of C3, which is a central and critical step in the complement cascade, also known as the common pathway. The common pathway is initiated when C3, a large glycoprotein, becomes activated. The activation of C3 results in the formation of C3 convertase, which are enzyme complexes. C3 convertases, in turn, further cleave C3 molecules into C3a and C3b fragments. C3b is crucial in opsonation, which is the process where it marks pathogens for phagocytosis, and in the formation of membrane attack complexes, also known as MACs. The C3b fragments generated in the previous step can associate with other complement proteins to form C5 convertases. C5 convertases cleave complement components C5 into C5a and C5b fragments. The C5b fragment, along with other complement components, including C6, C7, C8, and multiple C9 molecules, assemble into the membrane attack complex, also known as the MAC. The MAC inserts into the membrane of the target cell, usually a pathogen or infected cell, and forms pores, causing cell lysis and destruction. The activation of C3a and C5a fragments also have important inflammatory functions. These anaphylatoxins promote inflammation, stimulate mast cell degranulation, and recruit immune cells to the site of infection. Adaptive immunity, also known as specific immunity or acquired immunity, is a branch of the immune system that provides highly specific and long-lasting protection against pathogens. Unlike innate immunity, which is the body's immediate non-specific defense mechanism, adaptive immunity involves a more tailored and precise response. Adaptive immunity is characterized by the ability to recognize and remember specific antigens and mount a targeted immune response. Adaptive immunity is characterized by the production of antibodies and the activation of T cells. B cells produce antibodies that can specifically target and neutralize pathogens, while T cells can directly attack infected cells. When a B or T cell encounters its specific antigen, it undergoes clonal selection. This process involves the proliferation of the antigen-specific cell to generate a clone or effector cells, which carry out immune functions, and memory cells, which provide long-term immunity. One of the defining characteristics of adaptive immunity is its ability to form immunological memory. When the immune system encounters a pathogen, it can remember that pathogen, allowing for a more rapid and robust response upon subsequent exposures. This memory is the basis for the long-lasting protection provided by vaccines. B cells are a type of white blood cell that can produce antibodies, immunoglobulins, specific to particular antigens. These antibodies can neutralize pathogens, facilitate their elimination, or mark them for destruction by other immune cells. It does this through a series of a few steps. Lymphoblasts, which are B cell precursors, become activated into B lymphocytes. B cell memory is generated upon a first pathogen encounter and is carried by two distinct lineages: long-lived plasma cells that secrete protective antibodies for a lifetime, and memory B cells that can elicit improved and enhanced responses upon new antigen challenges. T lymphocytes are divided into two main types. These are known as CD4+ T cells, also known as helper T cells, and CD8+ T cells, also known as cytotoxic T cells. Helper T cells assist in the immune response by activating other immune cells, while cytotoxic T cells directly kill infected or abnormal cells. Major histocompatibility complex molecules present antigenic peptides to T cells, allowing T cells to recognize and respond to specific antigens. MHC class I molecules present antigens to cytotoxic T cells, while MHC class II molecules present antigens to helper T cells. In addition to these processes, the adaptive immune system has mechanisms in place to avoid reacting against the body's own tissues. This is known as immunological tolerance, and it prevents the development of autoimmune diseases. This process is carried out by regulatory T cells, also known as TREGs. TREGs can suppress the activation and proliferation of other immune cells, such as antigen-presenting cells. They do this through various mechanisms, including the secretion of immunosuppressive molecules like interleukins 10 and transforming growth factor beta. This has just been a very quick rundown of the immune cascade. If you would like to learn more, then as always, I will place all of my references in the description below. Thank you very much for watching.